In this section, we'll be looking further at the different types and strategies that we can use to inhibit enzymatic activity. So in this section, we'll be looking at several different ways we can regulate enzyme activity. Now we'll be looking at inhibition at allosteric binding sites. We'll also be looking at both uncompetitive and non-competitive inhibition. And then we'll finish up by looking at transition state analogues and suicide substrates. Allosteric binding sites offer a second target on an enzyme where a drug can have its mechanism of action. Now, binding of a molecule to an allosteric site will allow complete control of enzymatic processes, and binding at allosteric sites can either be reversible or irreversible. So we can have reversible or irreversible inhibition at these sites as well as at the active site within the enzyme itself. So what is an allosteric binding site? Well, this is our enzyme molecule, and here we have our substrate and our active site here. Now, the substrate needs to enter into the active site in order to be able to undergo a chemical reaction. Now, after it's done this, the substrate is then released. An allosteric inhibitor is one that doesn't directly compete with the substrate. Now, what it does is it binds to a different site on the enzyme. So this is a binding site other than the active site. Now, when the allosteric inhibitor binds to the enzyme undergoes a conformational change, and this alters the shape of the active site. Now, in doing this, it means that the active site is no longer able to accommodate the substrate, and therefore the chemical reaction cannot occur. Now, 6-mercaptopurine is an example of a molecule that binds to an allosteric binding site. Now, this molecule, which you can see here, binds to the first enzyme that is involved in purine biosynthesis. So this is the first enzyme in an enzymatic pathway that produces nucleic acid bases, such as guanine and adenine. Now, it blocks production of these purines and consequently blocks the production of DNA. But the 6-mercaptopurine binds to an allosteric site on this enzyme, so it doesn't compete directly with the substrate. It binds elsewhere and causes the enzyme to change its shape, and it's this that prevents the substrate from binding. As the substrate can't bind, you cannot produce the basic building blocks that you need for the production of purines, and because there are no purines available, DNA synthesis is then halted. This then prevents cell replication and eventually leads to cell death. Now, one of the major advantages with allosteric inhibitors is that because they don't directly compete with the substrate, the presence of the substrate doesn't matter, doesn't affect the binding of the inhibitor into its allosteric binding site. However, it also means that adding more of the substrate won't displace the allosteric inhibitor. So it means that if we were to give an overdose of the allosteric inhibitor, we can't correct this simply by adding more of the substrate, like we can with a standard competitive inhibitor. Next, we're going to talk about uncompetitive and non-competitive inhibitors. Uncompetitive inhibitors are inhibitors that bind reversibly, but they bind to the enzyme substrate complex. So when a substrate enters into the active site of the enzyme, the uncompetitive inhibitor will enter into a newly formed binding site and bind into that complex. Now, what this means is that the inhibitor does not compete with the substrate itself. So increasing the concentration of substrate will not affect the inhibition by the uncompetitive inhibitor. However, we need to make sure that there is enough of a concentration of substrate to ensure that the binding site for the uncompetitive inhibitor to enter into is formed. So we need to have a minimum level of substrate to ensure that there are enough substrate enzyme complexes for the uncompetitive inhibitor to bind into. Now, the uncompetitive inhibitor will only be able to bind and inhibit the enzymatic reaction when the substrate has already entered into the enzyme and makes this new binding site for the inhibitor to enter into. So it's very important that we have enough enzymes with substrate in their active sites to make this binding site available for the inhibitor. Now, non-competitive inhibitors bind to allosteric binding sites. Now, they bind and inhibit enzymatic activity, but they do not inhibit substrate binding. So what this means is that they change the enzyme in such a way when they bind that the enzyme is unable to complete the chemical reaction. So it's unable to convert the substrate into its products, but it doesn't stop the substrate from binding into the active site. It only stops the chemical reaction. Now, inhibitors that work in this way we call non-competitive inhibitors. Now, an example of this is the molecule nevirapine. Now, this inhibits HIV-1 reverse transcriptase through a non-competitive inhibition. Now, it binds to a binding site, so an allosteric binding site on the enzyme, which is close to the catalytic center, so it's close to the active site, but it's far enough away that it still allows the substrate to bind into the active site. Now, the binding of this molecule may alter the position of some of the key amino acids that are involved in the catalytic step that lowers the rate of reaction for the chemical reaction. So what it does is it changes the position of some of those amino acids, and that is enough to stop the reaction from being catalyzed. This means that whilst the substrate can still enter into the binding site, the chemical reaction doesn't occur. And so non-competitive inhibitors bind to allosteric binding sites and inhibit enzymatic activity, but do not prevent the substrate binding. They inhibit the activity by preventing the substrate from undergoing its chemical reaction, and therefore inhibit the production of products. And now we're going to discuss transition state analogues. Now, a transition state analogue is an inhibitor that has been designed to mimic the transition state in an enzymatic pathway. Now, what this means is that the transition state analogue has been designed to mimic the substrate around halfway through the enzymatic reaction. So it's been designed to mimic the substrate as it would look around halfway through undergoing the reaction that the enzyme is catalyzing. Now, what this means is that when the transition state analogue binds to the enzyme attempts to initiate the reaction, but the reaction is then stopped because of this inhibitor. Now, what this means is that the enzyme cannot complete its reaction and so will not release the product. In this case, it will not release the inhibitor. And so the transition state analogue will remain almost permanently bound, almost irreversibly bound to the enzyme, because the enzyme can't complete the reaction to be able to then release the inhibitor. So it should bind very strongly and give almost irreversible inhibition. Now, the development of renin inhibitors exploits exactly this approach. So renin is an enzyme that converts angiotensinogen, which is a peptide-coupled hormone, into angiotensin 1. Now, angiotensin 1 is a peptide which is then broken down to a shorter peptide called angiotensin 2. And this happens when angiotensin 1 is converted into angiotensin 2 by the angiotensin converting enzyme called ACE. Now, angiotensin 2 acts as a blood vessel constrictor and also retains fluid in the kidneys. And so it can be a cause of high blood pressure. Now, renin inhibitors mimic the transition state analogue of angiotensinogen when it's bound to the renin. And so what happens is the inhibitor will bind very strongly to the renin and prevent it from converting angiotensinogen into angiotensin 1. Now this means that the production of that is inhibited, and so we can't produce angiotensin 2. There's no angiotensin 1 for ACE to be able to turn over. And so inhibiting the renin will have the effect of being an anti-hypertensive agent. It will stop the hypertension because it inhibits this entire pathway. And this pathway results in the release of angiotensin 2, which causes blood vessels to constrict. So by inhibiting the renin using a transition state analog, we're able to stop this pathway from functioning and reduce hypertension. Now I'd just like to show you this example of how the renin inhibitors function, but you don't need to memorize this example. It's important to have an appreciation of what a transition state inhibitor is, but you don't need to memorize this specific example. Now the reaction that renin catalyzes is the breaking of a peptide bond. Now within this reaction we have two aspartyl residues. So these are aspartic acid, amino acid side chains here and here, and we also have a water molecule. Now all three of these components are crucial to the mechanism of action of this enzyme. Now what happens is the oxygen here removes a hydrogen from this water molecule, and then the remaining OH group is able to act as a nucleophile and react with the carbonyl in this peptide bond. Now this can then go on to remove the hydrogen from another aspartic acid side chain, and this then produces a tetrahedral intermediate. So the intermediate in the reaction is a molecule that has a tetrahedral arrangement around this particular carbon. Now what then happens is the reaction will go to completion via the oxygen on this aspartic acid removing a hydrogen from this OH group and allowing reformation of the carbon-oxygen double bond. This will break the peptide bond at this position. And you can see here we have the peptide bond broken, and the original two aspartic acid side chains have been regenerated. Now it's the tetrahedral intermediate here that is key to the design of the transition state analog. Now this transition state is a very high energy transition state. It's not particularly stable, and so what we can't do is isolate this transition state on its own. We need to rationally design a molecule that is based on what we know about this intermediate. Now this is what led to the production of this molecule here, which is called aliskiren. Now this is a transition state analog that is able to mimic a state that is close to the tetrahedral intermediate that we have here. Now this is the center that will be located in the same position when it enters into the enzyme as the tetrahedral intermediate. Now you can see that this is a carbon, it has an OH group on it, and it will also have a hydrogen group in this orientation coming forward, so out of the screen. So it is a chiral center. It doesn't have two OH groups because this makes the molecule very hard to synthesize, but it does have one, and it is in a tetrahedral arrangement. Now what we can see here is the transition state mimic, just the site that's important, and we have the reaction intermediate here. Now the reaction intermediate is tetrahedral. We have two OH groups, one of which is coming towards us and one away, and the unit here that is the transition state mimic only has one OH group, but it still has a tetrahedral arrangement around this carbon center. So we have a hydrogen that would be coming out towards us from the screen that isn't showed because this is a skeletal view of the molecule. Now the intermediate is closer in character to the transition state. So the intermediate that we have in this reaction is close to the transition state mimic that we have here. And this is what makes the tetrahedral design most suitable. Now this intermediate is able to enter into the enzyme and block its function. And the inhibitor is also resistant to further chemical reaction. You'll note that the inhibitor does not have an NH group here, it has a CH2. And that stops any cleavage at this bond here. So in the intermediate, we would have cleavage between this carbon and this nitrogen to generate our products, as shown up here. The transition state mimic has a carbon-carbon bond which is much stronger and won't be cleaved. So it means that the enzyme cannot process the analog and will not get through to undergoing the chemical reaction. It'll get stopped about halfway through, which is exactly what we want. We want the mimic to stop the reaction, and to do that it has to be resistant to any further reaction as well. So this aliskiren is the transition state mimic that prevents this reaction from happening. Once it's bound nearly irreversibly, and it stops the reaction from occurring. It stops the enzyme from turning over and stops this reaction taking place. And this is just one example of how transition states mimics or transition state analogues can function as inhibitors of enzymatic activity. Now we'll finish up by looking at the concept of suicide substrates. Now enzymes and substrates have some degree of flexibility in their interactions, so this explains why enzymes can turn over multiple substrates. Now drugs capable of forming covalent bonds with the enzyme are likely to act indiscriminately. So they are likely to act on not only the enzyme but also other enzymes or biomolecules that we may not want to react with. And so drugs capable of forming covalent bonds can react with a wide variety of targets, not just the targets that we want it to go after. And so ideally what we want is delivery of a drug that will only have an effect once it enters into the correct enzyme. And so this is a very attractive target. If we can deliver a drug which will only have an effect once the correct enzyme site is found, then this is a very attractive idea behind a drug, because it means that we can have very specific targeting of the enzyme by the drug molecule. And this is what the concept of a suicide substrate is based on. Now, suicide substrates are completely harmless molecules until they are converted within the enzyme active site. So they're molecules that are harmless until they find the enzyme, enter into the active site, and then are able to react with the enzyme that they're built to target. Now these will form a covalent bond to amino acids within the active site, meaning that binding is permanent, but that we can, with a very high degree of accuracy, make sure that this molecule only binds to our intended target and not to other proteins. Now here is an example, and you don't need to memorize this example. On the left we have pyridoxical phosphate, which can react with alanine. Now this reaction is catalyzed by the transaminase enzyme, and it will produce pyridoxamine monophosphate and pyruvic acid. But we can inhibit this by a suicide substrate called trifluoroalanine. And this molecule will then enter into the transaminase enzyme, and it will only react with the enzyme once it's entered into the active site. And so it will inhibit the production of the pyridoxamine monophosphate and the pyruvic acid. And just to show you the example of the reaction that takes place, now again, you do not need to memorize this reaction, but I just want to show you how this suicide substrate works. So here we have our pyridoxal phosphate, and we're trying to produce our pyridoxal amine monophosphate and pyruvic acid. So here we have our two starting materials. Now this starting material here can either be our natural substrate if these R groups are hydrogens, or they can be our trifluoroalanine if this R group here is fluorines. Now what initially happens is a condensation reaction to form an imine. So you can see here that we've formed a bond between the substrate and the pyridoxal phosphate. And what then happens is the enzyme, or rather a base from the enzyme, is able to come in and induce this rearrangement reaction here. Now this gives us this material, and if the R group, so the R's here, are hydrogen, this then allows us to move through and produce our product, our pyridoxamine monophosphate and our pyruvic acid here. However, if the R groups are fluorine, we move down a different reaction pathway. Now, this pathway, we have a rearrangement followed by irreversible alkylation. And so at this point, the product is fixed and we have a permanent covalent bond between the fluoroalanine and the enzyme itself. Now this bond cannot be broken and it holds the product in place here, and so it means that the enzyme is no longer able to have new substrates enter into the active site. And so what this means is that the enzyme is inhibited. Now the trifluoroalanine is not dangerous, it doesn't react with other enzymes because it relies on this enzymatic reaction. It relies on this pathway to generate our permanently covalently bound species. So what it means is if it were to enter into the active site of other enzymes, there wouldn't be a chemical reaction. And so the molecule would not be held covalently in place. And so it's an inhibitor that will only act very specifically on this particular enzyme. And this is the very concept of a suicide substrate. A substrate that is harmless until it enters the active site of the enzyme that it's been built to target, after which it forms a permanent covalent bond. Now, unfortunately, no system is perfect, and it is possible in some cases for suicide substrates to react with targets other than their intended target. So it can react sometimes with other molecules as well. And this was the case for the drug tianylic acid. Now, tianylic acid was a drug that was produced to be a diuretic, but it was eventually withdrawn from the market because of toxicity caused by it acting as a suicide substrate against cytochrome P450 enzymes. Now again, you don't need to remember any of this, you just need to be aware that suicide substrates can sometimes attack or inhibit the wrong target. But you can see this example here with the tianylic acid. When it approaches cytochrome P450, it's able to react with a cysteine amino acid here, or with a cysteine side chain. This is able to react with the molecule, and the molecule can then be a suicide substrate. So the enzyme becomes alkylated, it permanently has this group attached to it, and its mechanism of action is inhibited. Now this is an undesirable side product from introducing this drug into the system. This is something that we don't want to have happen. And so this drug was withdrawn due to the toxic side effects caused by precisely this mechanism of action. Now this brings us to the end of the section on enzymatic inhibition, and so you should be able to explain the role of binding interactions in the allosteric modulation of enzymes, and you should also be able to explain the molecular basis for a variety of enzyme inhibition processes. Now this includes reversible and irreversible inhibition, inhibition at allosteric sites, uncompetitive and non-competitive inhibition, transition state analogues, and the use of suicide substrates.